Okay, so in the interest of time, I think we should get started. Welcome everyone, and thank you for joining today's Coffee Science Seminar. I'm Owen Powell, and I'm going to facilitate today's webinar and introduce two visitors we have from Germany. And I will also be their point of contact during their stay here in Brisbane. So if you can't contact them directly after the talks, then feel free to reach out to me. And I'm a research fellow in the Centre of Crop Science and sit within the predictive agriculture theme at QAFI. So I'd like to start with an acknowledgement of country. The University of Queensland acknowledges the traditional owners and the custodianship of the lands on which we meet. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. We recognise their valuable and continuing contributions to Australian and global society. Before we kick things off, there's a little bit of housekeeping. Today's seminar is scheduled from 12 to 1, and it's a little bit different in that we have two speakers, so we're going to try and give them around 20 minutes to talk each, with 5 to 10 minutes of questions after each talk. And we'll have everything wrapped up by 1. And throughout the seminar, if you have questions, please place these questions in the Q&A tab that I'll be monitoring, not the chat function in Zoom. So it's my great pleasure to introduce our speakers today. They may be a little bit tired. I collected them from the airport at midnight last night, but I'm sure they're going to put on a good show. Lennard and Sven are both PhD students from Justus Liebig University in Giessen, which has a long-standing collaboration with QAFI and UQ. So it's really exciting to have them here. And so starts a new wave of collaborations along with the announcement of a new international research training group between UQ and JLU, which will be starting next year. So they both are from the Department of Plant Breeding and have backgrounds in quantitative genetics and predictive agriculture. And today, Lennard is going to be talking about some work he's conducted on phenomic selection. So over to you, Lennard, if you can start sharing your slides.
Yeah, thank you, Owen. All right, you see my slides?
If you can just, yep, we can see your slides now.
Okay, cool, thanks. So yeah, as you said, I will talk a little bit about phenomic selection in the structured hybrid canola population that we did during the last month. So, but before explaining you what is phenomic selection, let's just briefly go through genomic selection. I guess most of you are all familiar with that since it's a standard tool in plant breeding, also in animal breeding, of course, for the last at least 20 years. And yeah, the special thing is that we have a training set where we train with both the genotype and the phenotype, and with that we can predict the phenotype of an unphenotyped set based on the molecular markers. The only disadvantage is that although it's getting cheaper and cheaper every year, the costs of it are still quite high, as you have to extract DNA from all the samples and put them and send them for genotyping. So a possible alternative for that could be phenomic selection, where you use, instead of genomic molecular markers, you use the profile of near-infrared spectroscopy, which is a standard tool for a lot of crops. You routinely measure it to phenotype for metabolomic components. So for example, in canola, we do it routinely to measure for the content of oil, proteins, glucosinolates, and so on. And yeah, the idea is to, instead of using the molecular markers for the prediction, we use the profile of the NIRS of the spectroscopy. So for each wavelength here on a certain range, we get a value of absorbance, and with that we built a similarity matrix similar to the genomic relationship matrix in genomic prediction. And then, of course, the advantages would be that it's a very high throughput and cheap method. And depending on what kind of tissue you use, it could also be non-destructive. So if you use the whole seeds, you can run the NIRS analysis and you still have the whole seeds, and then you can plant them in the field without destroying them, as you would do for genotyping. The original paper, the original work that was done for this phenomic selection was from a group of from France. Vincent and Al did this 2018. So they were not the first one who tried to predict the yield and other traits with using NIRSprofiles, but they were the first one who did this on a genotype level. So before it was only done on a plot level, and they were the first who started building this similarity matrix, and therefore we are able now to do it on a genotype level. And they did it with wheat and poplar, and they got good prediction results for yield and heading date. So after that, especially in the last 2 years, a lot of other publications came out where people used phenomic selection, especially in wheat again and maize, also in soybean. Yeah, and it seems to work quite good. And the hypothesis why this works, so here's a nice figure by Charlotte Broad, who did a publication on grape wine. And so the thing is, for genomic prediction, we directly use the genome to predict the phenotype. And for phenomic prediction, we go through the whole cascade from genome over transcriptome, proteome to the spectra that we measure by the NIRS, and then we predict the phenotype. So by this, we still capture somehow the genomic variation of the plants, of the genotypes that we measure. And also at each step here, we capture environmental effects. And this is a hypothesis why this could work even better than genomic prediction. So yeah, we asked ourselves, would this also work with rapeseed? Because our group, we are mainly working, well, we're working a lot with rapeseed, and we had some data sets that we wanted to try phenomic selection with. So the data set that we chose was originated by we call it micronam population, where we had 5 different founder lines crossed with one common line here to get a population of 205 individuals that were then crossed again with 2 different mother lines, so that in the end we ended up with 410 different hybrids. The genotyping was done with the 205 father lines with a 15K SNP chip, and after aligning it to our reference genome and filtering it for several things, we ended up with around 6,200 markers for the prediction. The phenotyping was done on the hybrids in 4 different locations across Germany in the seasons 2019, 20, and 2021. And the field design was a PWeb design with 1.2 replications. The traits that we looked at here was, on the one hand, we used yield, as you can see here, for the different environments. We had a heritability of 0.28. Then we also looked at the plant height with a heritability of 0.24 and the flowering time. Before prediction, we used a linear model to adjust for environmental and field design effects that you can see here. So yeah, we basically, as random factors, we had the environment as well as the rows and column within each environment and the genotype by environment interaction. And we did this not only for the single traits, but also for each wavelengths of the NIRS profile to get one aggregated NIRS profile for each genotype, as we measured the NIRS also in each environment. So yeah, as you can see here, this is the original, the rare profile. on a range between 608 and 2,500 nanometers for wavelengths. Each 1 nanometer is 1 wavelength where we get the values. And after adjusting them for environmental and field designs effects, we also used the Sawicki-Golay filter to get rid of background noise and this shift. But this is a method routinely done with NIRSprofile. Yeah. Okay, the different prediction models that we used was, so to compare it with genomic selection, we used a GBLUP using a genomic relationship matrix calculated according to Van Raden. And then with the NIRSprofiles, we did a NIRSBLUP, where we calculated the similarity matrix by this formula here. So W is the matrix with the wavelengths times genotypes, and L is the number of wavelengths. And then we also tried to combine them in one model where we put both of the similarity matrix in one model. And then we also compared them with some other models like reproducing kernel Hilbert space regression, Bayesian lasso, and 3 different machine learning models, where we also had to tune the hyperparameters, and for that we used the Bayesian optimization approach. The first scenario that we tried was that we aggregated, like I explained already, the traits and also the NIRSprofile over all environments to get one aggregated profile for each genotype. And for cross-validation, we splitted the data set into 80% for training and 20% for prediction, and we did this 100 times with random splits. Now let's come to some results. As you can see here, for yield, the prediction using NIRS works better than using SNP. So here we have these 6 different models that we used, and the prediction accuracy is around 0.54 here for NIRS, and when we combine them both in one model, we even get some higher prediction accuracy. And this effect is even a bit higher for plant height, as we have lower accuracies here for the normal genomic prediction, but still for phenomic prediction we get higher accuracies up to 0.65, and they can still be a bit higher when we use the combination of both. On the other hand, here for flowering time, we already have good accuracies for genomic prediction, which cannot be beaten by phenomic prediction, but still with the combination it gets a bit higher. And the second scenario that we tried is, for now we use just one single environment to take to obtain the NIRSprofile. So one reference environment predicting still the traits of all environments. So here you see in each plot the environment, so for each plot, those environments that we used to train the model, the NIRSprofile from which we trained the model, and it still works quite good. And as you can see here, maybe it's easier to see it here where we used not the environment but the location, so we aggregated both years. What you can see here is that from those environments where the yield is on a level closer to the overall mean, those environments seems to work better to predict the overall aggregated yield of all environments. So for example, here, this environment and this, they are higher or lower than the overall mean, and this is why they are not that, the ability to predict the overall yield is worse here and better for those which are nearer, closer to the overall mean. Another thing that we were interested in was how the population structure affects the accuracy, because as you can see in this principal component analysis here using SNPs, the population is quite structured. Although here, when we did the PCA using the NIRSprofiles, we get one big cluster, just some individuals that are outside of this cluster. So what we did is family-wise cross-validation, where we used 4 out of these 5 subfamilies to train the model and then to predict the remaining subfamily, the yield of the remaining subfamily. So this is maybe a bit confusing, but still here we have the 6 different models that we used, and the symbols are those subfamilies that we predicted with the other 4. But let's focus on the overall mean here, overall predictions. And as you can see here, still for yield, the phenomic prediction and the combination gets better accuracies compared to the genomic prediction. The same for the plant height, and now interestingly also for the flowering time. So we conclude on that phenomic prediction is less influenced by the population structure compared to genomic prediction. And then one thing, in one of these 4 environments or locations, we also grew these 205 father lines. So we were really interested in if it is possible to also predict the yield of the hybrids or the traits of the hybrids by using the NIRSprofile of the fathers. So far, we did it just with the harvested F2 seeds of the hybrids, but of course, it would be more interesting to predict the next generation, the hybrids, using the seeds of the fathers. And we did this in 2 different approaches. First, we predicted only the traits in this specific environment, in this specific location where we also grew the fathers. And then we tried to predict the overall mean of all environments. And as you can see here, for yield, it works quite good. If we predict in the same environment, we still get on a similar level here, at least for the combination of both. And if we predict over all environments, it gets a bit worse, but still here for the linear model, we have accuracy on the same level as before with the genomic prediction. So this here for plant height, it looks quite similar, although it gets a bit worse for the model where we predict on over all environments. But here on flowering time, again, the linear model is on the same level or comparable level than with the genomic prediction. And also here, the combination looks quite good. Okay, yeah, so to conclude, we saw that phenomic prediction outperforms genomic prediction when using the F2 seeds of the hybrids to obtain the NIRSprofile for prediction. And as we saw in the combination, the combination got better prediction accuracy. So we conclude that genomic selection programs can be even improved by including spectral data. And yeah, we also saw that phenomic selection seems to be less influenced by population structure, as we did the cross-validation, the family-wise cross-validation, and using the parental NIRSprofile resulted in comparable good prediction accuracies compared to the genomic selection. Okay, with that, I would like to move my last slide. I would like to thank all my project partners and also Sven, who was involved, who contributed a lot to this work. And now I'm open for questions. Thank you.
Thank you, Lennard. Thanks for sharing your work. So we already have a couple of questions in the chat, so I might just start straight away. So we have a question from Scott Chapman, and he's interested in how the near-infrared phenotypes were measured. So he's asking, are they measured on each plot and at what frequency through the season?
Yeah, so the NIRSprofile, they were measured in the lab after the harvest by using the whole seeds of the plants. So we harvested the plants, and then for each plot, we took a couple of seeds, measured them in the lab like we would routinely do to a phenotype for protein and oil content, for example. So we did this already, we had the data, and these were then, the profile were then used for the prediction. Okay,
thank you for clearing that up. Yep, okay, Scott's happy. We have another question from Graham Hammer. So he said, nice black box sort of approach, but do you sort of have any thinking about what the biological rationale for spectral reflectance of seed relating to traits such as yield and height? So can we dig into any of the biology?
Yeah, so that's a very interesting question where we don't have yet a proved answer, but I mean the theory is that with the spectra we capture metabolomes, like the metabolome, like for example protein, as I said, or oil, and these are somehow correlated to, for example, yield. And maybe with this correlation the models are able to predict yield in this case. And like I showed in this slide, we have this whole cascade from the genome over the transcriptome to the spectra. So we somehow capture the genomic effect or the genomic relationship between the individuals in the spectra, and this could be somehow captured by the models for prediction. Yeah.
Okay, so that leads nicely onto a question I have. So if you're sort of indirectly capturing some of that underlying biology, do you think that's going to be stable across environments? So from your slides, it looked like you corrected out for environments before you used the spectral data. So have you thought about how you would more explicitly actually have G by E in there, which you mentioned as a possible benefit?
Yeah. So like you said, so far we corrected for genomic for environmental effects before we trained the model, but this is one thing that we would like to try in the next step, to have a combined approach where we directly do the prediction and include G by E effects in the prediction model to see if this will have an effect. Yeah.
Perfect. I think there are a couple more questions, but on a related note. So in the interest of time, I might leave them and point those questions to you after the seminar, Lennard, that you can follow up with individuals, if that's okay everyone. And we can introduce the next speaker. So Lennard, if you can push on the slides, thank you. So next up we have Sven Weber, who's also a PhD at the Plant Breeding Institute. A similar background to Lennard, but he's going to be talking about some work using 3D point clouds, again in canola, I believe. Sven can correct me. Take it away.
Great. Hello. Thanks for the nice introduction. Other than stated in my screen, I'm not Lennard, I'm Sven, and I'm going to talk a bit about 3D point clouds for high throughput phenotyping in canola, which is only a small part of a bigger project to increase N efficiency in canola through extension of genetic diversity. So as a small introduction, I just want to briefly mention the hypothesis of the whole project. So the idea was that the extension of the genetic diversity also enriches the variation for nitrogen use efficiency in breeding pools. And then the second hypothesis is that plant architectural traits may have higher heritability than yield or N use efficiency and are correlated to those. And by assessing those architectural traits, we can kind of passively select for N use efficiency and seed yield. And then the third part, where I was mostly involved last few weeks or months, is that the digital assessment of plant architectural traits can be like a useful tool for industry and for science in plant breeding. So I want to lay focus in that talk today on the last two hypotheses, because I was most involved there and I find them the most interesting. So starting this off with kind of describing the population that was created in that project. So we started off with 40 genetic diverse founder lines, which are basically pre-breeding lines. So very, very pre-breeding. and they were used to create a NEM population. And from that NEM population, we selected genotypes that have a relatively high yield in certain environments and are genetically diverse based on genetic diversity or based on Rogers distance. And we were left after the filtering and selection with roughly with 160 genotypes. And these 160 genotypes were used to produce hybrids with 2 different hybrid mothers. So in total, we had 320 hybrids, which were subsequently tested in the fields. Coming to the field trials where we tested all those hybrids, so they were scattered all over Germany. One thing I want to mention here especially is the locations from our university. So we have one location near Giessen itself and one location in the south of Frankfurt. But in total, we had 7 locations across 2 years, so 14 environments. And since it's an end-use efficiency project, we set a relatively low N fertilization of 125 kilogram per hectare nitrogen, which is in Germany quite low. So farmers usually fertilize up to 200 kilos in Germany for canola. So by that, we passively assess the end-use efficiency through seed yield, and the field design was a PREP design. So after all the data was collected for yield and all phenotype data, we used a mixed linear model to extract means with for the genotypes as fixed effect. And now coming to the nice parts, so the plant architecture in canola. So the architecture is quite complex, I would say, in canola compared to, let's say, wheat. And we only assessed architectural traits in one environment near Giessen because it's a huge effort and it's laborious intense. And we assessed many traits. So we assessed, for example, plant height, pod number, number of side branches, pods per side branches, branch angle, all stuff like that. So it was a huge effort. So we had, let's say, 15 technicians working on that every day for 14 days straight. And the way we did it was we sampled 5 plants per plot and assessed all the traits on each plant individually to have a representation of the whole plot. So coming to the first results here from all environments again for yield, what we do see here is quite some variation across all the NEM families on the x-axis. And 2 things I want to specifically show here is that it seems that mother hybrids originated from the mother B in yellow seem to have generally higher yields compared to the mother A in blue here. And the second thing is that most hybrids seem to not reach the level of a commercial elite hybrid. Only very few reach that level, which is by nature, I think, based on the very diverse material that was used there from pre-breeding. But as I said, architecture plays a big role in that project. We have here one architectural trait. So this is pods per side branch on the second side branch. Don't mind the German word for that on the y-axis. So it's the second side branch, it's the pod number there. And we again see the contrasting effects between the 2 mother lines, but we don't see a huge difference, or we don't see a difference to the lead material. So we have the NEM population is on a quite similar level, at least for those branches. But I said we did it not only for one side branch, we did it for all of them. And what was really interesting for us is we have a gradient across the plant. So starting from the lowest branch, the pods on that branch, the pod number doesn't really correlate that well to the important trait hybrid yield. But as you go a bit higher to the middle part of the plant, the number of pods per branch starts to correlate higher and higher till you get to that middle part here with yield. And then going wider up or more up to the top end of the plant, you see that the correlation increases again. So we conclude on that the middle part of the plant, or the pods on the middle part of the plant, seems to play a large role for seed yield compared to the upper and lower extremes. So what anyone usually does with such trait is an association study. Here we found the highest correlation on the 6th side branch to yield here on the 6th side branch, and that's why we choose to do an association study there. And what we see here is that you may find some QTLs on A10, chromosome A10 or C8, but no like huge QTL with high lot scores. So we conclude on that we need to better describe the genetic effects, and doing so we need to have a bigger population characterized for those architectural traits. But as I said earlier, it's quite laborious intensive. So we did like 2,000 plants in 1 year, and it was such an effort. So it's not feasible to go even higher. So we asked ourselves, can't we do that in a better way digitally without all the involved work? So what we did is while we were doing the phenotyping by hand, we scanned each plant with a kind of LIDAR scanner or a 3D scanner from Phenospecs. It's the plant eye. If you want to know it in detail, you can ask me later, which basically kind of creates a so-called point cloud. So you see here on the right side, the output of this 3D scanner looks basically like that, and you have plenty of points representing the plant. So the issue here is we wanted to use those point clouds together with machine learning to phenotype automatically. But the issue with point clouds really is you only get the table with X, Y, and Z axis and the according points, but all models that are usually used rely on very structured data, but these aren't structured, because if you like, for instance, change 0.2 to 0.5 or the labeling, the point cloud will remain the exact same thing, but any other algorithm won't be able to understand this exchange. So you need to be invariant to that order, and then the point of view shouldn't matter because the plant remains the same independently from where you look at it. So it always has, for instance, like 100 pods independently if I look from the top or from the side or from whatever. So this is also a problem of those point clouds. And the way we kind of wiggled around this is we basically took pictures from multiple perspectives of the plant and used those pictures as a kind of picture 3D representation of the true point cloud. So each picture was then used to train a so-called CNN, convolutional neural network, which are basically very, very good at classifying or regression tasks on pictures. And we trained basically this CNN on all the perspectives of one plant, and then basically pooled the perspective with a kind of view pooling. And then on that pooled view, we did a regression task. And if you are interested in more detail, it's called multi-view CNN in literature. So this was the way we tried to train a model to predict those traits. And here you see the results of that from tenfold cross-validation. On the y-axis, you have the correlation, and on the x-axis, you have the traits. And what you can really nicely see is that it's working quite well for the total number of pods per plant. It's not working so well for pods on the main branch, but again, it works quite well on the pods on the side branches. So we think that it's mainly driven here, the total pod number accuracy, by the number of side pods per side on the side branches. Sorry. Where it didn't work that well was on the number of side branches and the branch angles. So we are still at the process of benchmarking the models and optimizing the models. So we are quite optimistic that we can improve a bit on that still. So one question we asked ourselves, which side data we commonly assess could be useful to improve this prediction accuracy. And we commonly fly over our trials with an UAV, with a multispectral camera. And we know that some vegetation indices are highly correlated to, for instance, yield. So one result I want to show here in regard of that is the results based on the normalized difference in red edge. So here in that heat map, you see each square represents one genotype ordered by yield, and the color represents the NDR. So we see a quite nice correlation between yield and the NDR in flights before flowering, and then after flowering we still see that nice correlation, but it kind of shifts in the other direction, going to a negative correlation. So this showed us maybe there's something to capture in the UAV pictures. So we started to incorporate do the same model with also the UAV pictures from each individual plot here in yellow now. So the light blue is still the point clouds. Yellow is now based on UAV pictures. And what you see is that mostly you don't see a good result for the models, for the traits where it worked good on point clouds. You see some improvements for pods on the main branch, and maybe some improvement of the number of side branches, but nothing majorly. And then we tried, when we combine both models into one model, can this improve, or will this yield some synergetic effects? But unfortunately, for most of the traits, it wasn't the case, but we do see some improvements for the total number of plots, which can be could be due to the improvements for the accuracy in pods on the main branch, but nothing mentionable for all the other traits. But the main interest of a commercial breeder is, I would say, increasing yield in general. So we tried to predict yield, plot yield, with all these three approaches I just showed here. So we tried to predict plot yield. And what we did see is that it already at least correlated a bit with point clouds. So we were quite lucky that we find at least something, especially because you only capture plant architecture and not plot density, for instance. And then with UAV pictures, you are able to predict even better. And then if you include the point clouds and the UAV pictures, we get quite, I would say, good prediction results, which we, or which I would say are partly at least due to the incorporation of kind of plant density in the plots, and then the other big part is the plant architecture, like the plot number. So yeah, these are the first results for our 3D based phenotyping approaches together with UAVs. And the next thing here, which we plan on doing, is also incorporate genotypic data, but we are just starting on that and hopefully have some results in a few weeks. So with that, I would conclude that these 3D point clouds can be a useful tool for phenotyping, but we are just at a starting point, so there's still much room for improvement. And now I want to say thank you for your attention and want to ask you for questions. And yeah, this is it from my side. Thank you.
Thank you, Sven. I know there's a lot of people working in a similar space, so I'm sure there'll be lots of questions or advice on how they've tackled these problems. So I'll start with the question. So when you generate these 3D point clouds, do you take multiple samples per plot? So have you looked at the variation in those pod characteristics within a plot?
Yeah, so we sampled, or we used 5 plants per plot. But I have to admit, this was not my part of the project to look at the variation, or we didn't do it yet, to look at the variation between the different scans. But I would, as far as I remember, at least the levels were similar. I mean, one plant isn't explaining the other plant, but if the genotype has many pods on the side branch, all the scans had relatively high plots on the side branches. Yeah.
Okay, thank you. So we have a question from Professor Graham Hammer here. So he was wondering, did you compare looking at those architectural traits versus just looking at grain number at harvest? Because I think Graham's saying that a lot of this will just be yields correlated to pod grain or number. So do we have to go to all the effort of all these other characteristics?
Yeah, this is a really good point, and we're just checking on that. I think what's even more than just seed number here is the pure amount of biomass that consists out of plots. So the easiest answer for the model would just simply look at how much area is in general black in the picture, let's say it like so, but we didn't check this yet. But a great point, yeah.
Looking for any more questions? We've got a bit of time, so I'll ask one more. So you mentioned this problem of having to convert these point clouds for the algorithms to images. Can you maybe explain a bit more about that? And it sort of seems like you go to all of this effort to get point clouds, and then you just sort of take the images of the point cloud. Seems like a lot of work to shrink your data down.
It's hard because I can just quickly move to that slide. So as I said, order doesn't matter anymore. So you have to find some sort of algorithm which is capable of that, or some formula or some function. Yeah, it's a function. And what people have been trying is to find a function which is invariant to order, and the basic functions like mean and max and minimum are invariant to that, and there are models capable of using that idea. And we also tried those, but what we did find is that they don't work that well on our tasks. Yeah. Maybe that, and yeah, well, also the orientation, the point of view, it's really hard to work with point cloud because of that unstructuredness in the data. Yeah.
Okay, thank you. So I think we've hit a plateau on the questions. So I'd like to thank everyone for joining today's seminar and thank the speakers for sharing their work. Both Sven and Lennard will be around for the next couple of weeks, as well as a couple of their colleagues. So please reach out. I think they're looking to discuss their work and share their scientific ideas together. So please get in touch. Before we close the seminar, I just want to point that next week we have Kieran McCosker from the Centre of Animal Science presenting on the effect of shade on cow behaviour and calf survival in the Northern Territory. And if you require any information or to sign up for regular updates on the Coffee Science seminars, please follow the Coffee website /science seminars or contact one of the seminar committee team. And with that, I'll thank you all for joining and thank our speakers and have a great day. Thank you.